Welcome to the deep dive. Today we're getting into the secret language of life itself, how our cells talk to each other. It's called cell communication, or CCC.
It's fundamental biology. Yeah.
And we've got this amazing technology, single-cell RNA sequencing, scRNA-seq, that lets us listen in like never before. We can see exactly which genes are switched on in, like tens of thousands of individual cells, incredible detail.
Right. The resolution is amazing.
Yeah.
But there's a big bet.
Exactly. We can get the data, but actually understanding the conversations. We're kind of dropping the ball.
That's the core issue the sources point out. Almost all the tools we use for CCC analysis, they're what we call agglomerative.
Agglomerative, meaning?
Meaning they don't look at cell A talking to cell B. No, they lump cells together.
Yeah.
All your T cells in one bucket, all your fibroblasts in another. Then they analyze the average chat for the whole group, the cluster.
Okay, so it's like trying to understand city gossip by just looking at the average noise level in each neighborhood?
Precisely. You completely miss that one specific, maybe super important conversation happening between two really unique cells. The conversation that could be key for, I don't know, a disease process or something.
You lose the specifics. The crucial details get washed out in the average.
Exactly. That aggregation obscures the heterogeneity, the real variation. So that brings us to what we're diving into today. Scriabin.
Scriabin, single-cell resolved interaction analysis through binning. Sounds promising.
It is. It's a framework designed specifically to maintain that single-cell detail. No averaging, no statistically questionable downsampling. It keeps the focus on the individual cell pairs.
All right, so our mission for this deep dive, unpack Scriabin. How does it work? How does it handle huge data sets? And crucially, what hidden biology does it actually reveal?
That's the plan. We'll look at its core workflows and show some examples where it found communication networks that were just invisible before.
Let's start at the beginning then, the basic unit. How does Scriabin actually score the interaction between one single sender cell and one single receiver cell? Okay, let's get into the nuts and bolts. A sender cell makes a ligand, a receiver has the receptor. How does Scriabin turn that specific pair's gene expression into a number, a score?
Right, so they build this potentially enormous data structure. We call it the cell interaction matrix, or CCIM. And for each possible interaction between cell I and cell J.
Every single pair.
Every single pair, yes. They calculate a score using the geometric mean. It sounds fancy, but it's actually quite elegant.
Geometric mean. Refresh my memory.
Sure. So first, you take the expression level of the ligand in the sender cell, let's say $1, and the expression of the receptor in the receiver cell, no J. You multiply those two numbers together.
Okay, ligand times receptor expression.
Exactly. Now think about it. If the sender isn't making the ligand, expression is 0. Or the receiver doesn't have the receptor, expression is 0. What happens to the product?
It's 0.
Right. The score is 0. That's your biological AND gate. Both things need to be present for a potential interaction. No ligand or no receptor means no conversation possible. Score is 0. Done.
Makes perfect sense. Gotta have both sides of the handshake.
Precisely. Then there's a second step. They take the square root of that product.
The square root. Why?
It's basically a way to control the volume, like you said before.
Yeah.
Imagine you have one ligand-receptor pair where both genes are just expressed at astronomically high levels. If you just use the raw product, that one interaction could potentially dominate all the analysis just because it's so loud.
Ah, I see. So the square root dampens that effect. It stops the few really high expression pairs from drowning out potentially important but lower expression interactions.
Exactly. It prevents those outliers from having a disproportionate impact. So you end up with this matrix, encoding the interaction potential for all cell pairs across all relevant ligand-receptor pairs.
Okay, but you said potential. And if we're talking modern single-cell data sets, tens, hundreds of thousands of cells, that CCIM matrix must be absolutely gigantic. Millions, maybe billions of potential interactions.
Oh, easily. Computationally terrifying, yes.
So how do we know which of those millions or billions of potential conversations actually, do anything? Which ones have a real biological effect? Are we just creating a different kind of noise problem?
That's the crucial next filter. Just because cell A could talk to cell B doesn't mean that conversation actually changes cell B's behavior. Scriabin addresses this by weighting those scores. It prioritizes biologically active ligands.
Active ligands.
Meaning ligands that are predicted to actually influence the downstream gene expression programs inside the receiving cell. It asks, did this potential interaction likely cause a real change in the receiver?
Okay, how does it predict that?
It leverages other computational tools. First, it uses something called cell ID to figure out a unique gene signature for each individual cell. What makes that cell tick right now?
So a molecular fingerprint for every cell.
Sort of, yeah. Then it brings in another tool called NicheNet. NicheNet has this curated database and model that predicts which ligands are most likely responsible for driving the observed gene signature in a target cell.
Ha. Okay.
Uh-huh.
So the geometric mean says these two could talk. Then cell ID defines the receiver's current state, and NicheNet looks at that state and asks, okay, which incoming signals, which ligands are most likely causing this specific state?
You got it. If NicheNet points to a specific ligand as being highly active, likely driving the receiver's gene expression, Scriabin then increases the weight of interactions involving that specific ligand.
So it turns up the volume on the conversations that seem to be having a real impact.
Exactly. It refines that massive potential matrix into a map of likely functional communication.
Okay, quick recap. We have this core idea. Calculate interaction potential for every cell pair using the geometric mean, which ensures both ligand and receptor are present, and then weight those interactions based on whether the ligand seems to be actively changing the receiver cell's gene expression using tools like NicheNet. Makes sense. But you mentioned that full matrix, the CCIM, is just computationally infeasible for the really big data sets we have now, Atlas-level stuff. So how does Scriabin scale up without resorting to just averaging everything together, which we know kills the signal, or subsampling, which is statistically problematic?
Yeah, that's the scaling challenge. They offer two main ways around this, two workflows that avoid those pitfalls. The first one, let's call it workflow 2, is built around binning. This is especially useful when you want to compare samples like healthy versus diseased tissue.
Binning. Like putting things into categories. Isn't that a form of aggregation?
But it's a very, very high-resolution, carefully controlled form. First, instead of building the full detailed CCIM right away, they compute something simpler, the summarized interaction graph, or CL dollars. This just calculates the total magnitude of interaction between any cell pair, summing up all the individual ligand receptor scores. It's much faster to compute.
Okay, a summary score first, then the binning.
Then the binning. Imagine you have cells from sample A, healthy, and sample B, diseased. You can't just compare all T cells in A to all T cells in B, because the T cells themselves might be different or the proportions might change. Binning is a way to precisely align the cells across the samples.
How does it do that alignment?
It looks for mutual nearest neighbors, MNNs, across the datasets. Think of them as like transcriptional soulmate cells in sample A that are extremely similar to specific cells in sample B. These MNNs act as anchors.
Okay, anchors between the data sets.
Right. Then it assigns every cell to a bin identity. The goal is to create bins where the cells within a bin are as similar as possible, and importantly, each bin has cells represented from all the samples you're comparing. It minimizes aggregation while maximizing comparability across conditions.
So instead of comparing crude P-cell cluster 1 across samples, you're comparing bin 42, which represents a much more specific conserved cell state that exists in both healthy and diseased samples.
That's the idea exactly. High-resolution registration. Once you have these bins, you can ask, which bins show the biggest change in their communication patterns between the conditions? Which specific cell phenotypes change their interactions the most?
And then you build the detailed matrix.
Only then. You focus just on the cell pairs belonging to those high-variance bins, the ones where the communication seems to be changing significantly. It's like intelligent subsetting, guided by the comparative analysis. Much more manageable.
Clever. That handles comparisons between samples. But what if I just have one massive data set, like that fetal gut atlas example with 76,000 cells? I'm not comparing conditions. I just want to find the main communication pathways within that single huge data set.
Right. For that scenario, they have workflow 3, interaction program IP discovery. This one scales differently. The core idea here is shifting the problem. Instead of focusing on potentially billions of cell pairs, you focus on the types of interactions happening.
Types of interactions.
Yeah, think of it like finding recipes. Are there groups of specific ligands and receptors that tend to be used together over and over again by cells that are themselves transcriptionally similar? They adapt a method called WGCNA, which is normally used for finding co-expressed genes, but they apply it to find co-expressed ligand-receptor pairs.
So finding modules of communication signals that act like a coordinated unit.
Exactly. They call these modules interaction programs, or IPs. It's like discovering the common communication protocols or blueprints being used in the tissue.
And why is that scalable?
Because The number of potential ligand receptor pairs is fixed, defined by biology and the database you use. It doesn't really grow much whether you have 10,000 cells or a million cells. The complexity lies in finding the patterns among these pairs, not in iterating through every single cell interaction.
Ah, okay. The search space is the set of LR pairs, not the N by N cell matrix.
Precisely. Once you identify the significant IPs, these common communication blueprints, you can then just score each individual cell based on how strongly it seems to be using or receiving signals from each particular IP. It gives you structure and insight into huge data sets without needing that full initial CCIM.
Okay, this is where it gets really interesting, the payoff. We've talked about the how, the mechanics of CCIM, binning, and IPs. Now let's see the why. What actual biology did this single-cell approach uncover that the older aggregate methods were missing? You mentioned a stark finding about the cost of aggregation.
Yeah, the benchmarking they did was pretty damning for aggregation methods. They simulated ground truth data where they knew the real communication patterns. Then they tested how well different methods recovered that truth.
Yeah.
And when they artificially aggregated the data even just a little bit, like averaging within small neighborhoods, nearest neighbors, or using standard suitable clusters, the ability to find the true communication states got consistently worse.
Worse? How much worse?
Significantly. At nearly every level of aggregation they tested, they lost at least half, 50% of the unique, true communication states present in the original single-cell data.
50%. That's huge. So aggregation isn't just simplifying, it's actively throwing away potentially critical information.
It's signal obliteration, basically, which leads directly into the examples. Take T-cell exhaustion in squamous cell carcinoma, SCC. This is a really hot area clinically because exhausted T-cells are why immunotherapies often fail.
Right. The T-cells just get worn out fighting the tumor.
Exactly.
Yeah.
But the thing is, in real tumors, T-cells don't just flip a switch from active to exhausted. It's a continuum. They exist on a spectrum. In the SCC dataset they looked at, the exhausted T-cells didn't form their own neat little cluster on the standard analysis maps. They were kind of smeared across different T-cell groups.
So an agglomerative method, which needs to define an exhausted T-cell cluster to analyze its communication, would likely miss them or average them out with non-exhausted cells.
Precisely. It wouldn't see them as distinct communicators.
Yeah.
But Scriabin, working at the single-cell level,
It saw the difference.
it did. By calculating that CCIM, looking cell by cell, it found clear differences. The T-cells that were phenotypically exhausted were predominantly using inhibitory signaling molecules like CTLA-4 and TIGIT to talk to other cells. Meanwhile, the non-exhausted T-cells, even if they look similar on the overall map, were using things like pro-inflammatory chemokines, CCL4, CTL5.
Wow. Same apparent type on the map, but completely different communication strategies revealed only at the single-cell level. That has direct implications for understanding therapy resistance.
Absolutely. It makes the functional state visible through its communication.
Okay, another example you mentioned showed the danger of aggregating not just cells, but entire samples. The leprosy granuloma study, comparing two different forms of the disease, LL versus RR.
Right. So an older agglomerative analysis of that data suggested that ligands like IL-1B and CCL21 were the most upregulated signals driving the stronger immune response seen in the RR form.
Okay, seemed plausible.
Plausible, but wrong, or at least misleading. When they reanalyzed using Scriabin's binning workflow, remember, the one that carefully aligns cells across samples?
Yeah, the high-resolution registration.
It showed that the apparent high IL-1B signal wasn't a general feature of RR granulomas. It was basically being driven by one single RR sample that just happened to have a lot of T cells expressing IL-1B. It was an outlier skewing the average.
The classic outlier problem, masked by aggregation. So what did Scriabin find instead?
By using the bins to compare equivalent cell states across all samples, it correctly identified a specific subset of T cells marked by the gene CRTAM that were significantly upregulating interferon gamma, IFNG, specifically in the RR granulomas.
Interferon gamma. That rings a bell in leprosy.
It's biologically known to be a key driver of that RR state. And Scriabin showed these IFNG-producing T cells were signaling to myeloid cells, triggering them to produce the actual pro-inflammatory cytokines like IL-1B, TNF, CCL3. It found the real upstream driver, the IFNG signal, that the aggregated analysis had missed because it was swamped by an outlier.
That's a powerful correction. Okay, and what about sheer stale? The Fetal Gut Development Atlas, over 76,000 cells. They use the interaction program workflow there, right?
Yep, Workflow 3. And it successfully pinpointed a key communication module, they called it IP1, that seemed crucial for maintaining the niche for intestinal stem cells, the ISCs.
The stem cell neighborhood, basically.
Exactly. Now, this IP1 module confirmed the importance of a known factor, RSPO3, which helps maintain these stem cells. But here's the cool part. In mice, the standard lab model, RSPO3, is thought to be produced mainly by lymphatic endothelial cells, or LECs.
Okay.
But in this human fetal gut data, Scriabin's single-cell IP analysis showed RSPO3 wasn't coming from LECs. It was primarily coming from a specific type of fibroblast marked by the gene GREM1.
So a different cell type is doing the same job in humans compared to mice?
Seems like it, yeah. A species-specific difference in how this critical stem cell niche is maintained. And that's the kind of subtle cell-type-specific detail you'd likely miss if you just aggregated all fibroblasts or all endothelial cells together. The single-cell resolution was key.
We've covered interactions in space within a tissue, between conditions. What about interactions over time? How does keeping single-cell resolution help track a sequence of events?
Yeah, this is where the concept of longitudinal communicative circuits comes in. It's about connecting the dots temporally.
Okay, how does that work?
It tries to define a chain reaction. So you have sender 1 talking to receiver 1 and time point 1. That interaction, sinusoidal right arrow 1, needs to predict, based on NicheNet, for instance, that 3.1 will likely upregulate some new ligand, let's call it LA.
Okay, the first interaction primes the receiver to become a sender itself.
Exactly. Then, at the next time point 2, you need to find a cell, TS2O, that is transcriptionally very similar to 1R$1. They likely share the same high-resolution bit identity we discussed earlier. And this TS2 must then use that ligand LA to signal to a new receiver, 2.2.
So $2 right arrow R11, which becomes a $2 right arrow R22, linked by the predicted upregulation and subsequent use of ligand LA. It's like a molecular relay race across time.
That's a great analogy. It stitches together the potential flow of influence. And they use this to look at SARS-CoV-2 infection in human bronchial epithelial cells, HBECs, grown in the lab. They infected the cultures and tracked them over time. The question was, who kicks off the immune response?
The infected cells, presumably?
That's what you'd intuitively think. But Scriabin's circuit analysis found something really surprising, especially in the early phase, like day 1 post-infection.
What did it find?
It found that the initiators of many of these early inflammatory circuits were often uninfected bystander cells.
Wait, the cells that weren't infected were starting the alarm?
Yeah. Specifically cells like basal cells, ciliated cells, club cells in the culture. For example, a key early inflammatory signal, IL-1B, was often initiated by these uninfected bystanders, signaling to other cells, including infected ones. Same for other early signals like SAA1 and CTGF.
So the cells yelling fire first weren't necessarily the ones actively burning?
That seems to be the implication here. These non-productively infected cells might be crucial early triggers, sensing danger nearby perhaps, and initiating pathways that maybe get amplified later by the truly infected cells.
That completely changes the picture of early viral response. If you were only looking at virus-positive cells using an aggregate method, you'd totally miss that the initial push might be coming from their uninfected neighbors.
Exactly. It highlights how this single-cell longitudinal view can really challenge assumptions.
Okay, fascinating stuff. But we should also touch on limitations, right? No tool is perfect.
Absolutely. Critical thinking is key. Scriabin has some limitations noted in the sources. For one, it relies on existing databases of ligand receptor pairs, and it generally assumes they're all equally valid and function simply. It doesn't easily handle, say, receptors that need multiple different protein subunits to work.
Right, biology's messy.
It is. Also, because it often relies on NicheNet for predicting activity, it's currently better at finding signals that activate pathways in the receiver, rather than signals that inhibit them. NicheNet is focused on activation.
Okay, so it might miss some important stop signals.
Potentially, yeah. And the big underlying assumption for almost all these scRNA-seq-based methods, including Scriabin, is that the mRNA levels we measure are a good proxy for the actual protein levels doing the signaling.
Which we know isn't always true. Post-transcriptional regulation is a thing.
It's definitely a thing.
Yeah.
So interactions driven purely by changes in protein availability or modification without a corresponding mRNA change could still be missed. That's a general field limitation, really.
Good points to keep in mind. So wrapping this up, what's the big takeaway for someone listening? Why does this single-cell approach with Scriabin matter?
I think the main message is that Scriabin, by preserving that single-cell view, gives us this incredibly powerful magnifying glass. It lets us see the intricate structure of how cells organize and influence each other in their niches finding biological signals that are just fundamentally lost when you average things out.
It moves beyond just asking which cell type is talking.
Right. It lets us ask which specific cells are talking, when are they talking, and what are they saying. It helps explain why two cells that look almost identical on a standard cluster map might behave completely differently, because their communication patterns, their active signaling, is distinct.
Like those exhausted T cells.
Exactly, or those bystander cells in the SARS-CoV-2 example. Think about that finding. The initial inflammatory signals weren't necessarily from the disease cells themselves, but from their neighbors.
That leads to a pretty provocative thought, doesn't it?
It does. If the initiation of important biological processes, maybe disease onset, maybe even normal tissue maintenance, is often driven by these cells that seem phenotypically ambiguous, the ones that don't fall neatly into disease clusters, the bystanders. Well, what might we be completely misunderstanding about existing diseases?
Because our tools have historically been biased towards looking only at the obvious clustered players.
Precisely. What fundamental processes in health or disease have remained hidden simply because we've been averaging away the subtle single-cell conversations that might actually be running the show. And that's the really exciting frontier this kind of single-cell resolution approach opens up. What gaps can we fill next?